Okay, now you see the pictures we've just been looking at, all matched up with the much easier to see diagram. This is why you make diagrams. Things can be much clearer than they are in reality, so to speak, right? This cell has 4 chromosomes total. If this was your or my cells, there'd be 46. But that would be such a messy diagram that in a textbook, you're almost always going to see 4, maybe 2, maybe 6 at the most. So that's why you see that number. They just chose that to make this clear. Now notice something that's gonna be real important in a little bit. Notice that in this group of 4, there's 2 that are the same size and another 2 are the same size. That's going to be absolutely critical coming up next and for the entire next unit. So I'll save that for later. Okay, but here you don't see any chromosomes, right? So we're going to call that interphase, more specifically G2. Now you see some microtubules forming, right? The spindle's starting to form. You see chromosomes, but they're not lined up yet. They're not all the way coiled up. So if you want to call that early prophase, you can call it that. Here, they decided to call this early metaphase. Very confusing, right? Whatever. This is not identified as metaphase in your tests. You're going to see them lined up right along the middle if they're metaphase. So if they're not quite lined up on the middle, I would call this late prophase. This is clearly metaphase, right? Very clear in the diagram. This is clearly anaphase. And then again, this is clearly telophase because you see this pinching in the middle, which we're going to get a name in a second. That means, again, cytokinesis has started. At the end of mitosis, cytokinesis is starting. And then you see something else which I ignored before. The nuclear membrane, which used to be surrounding these chromosomes, right? Back in prophase, when this was just starting, there was a nuclear membrane. It's gradually gone away so that there's no nuclear membrane here or here. But now another sign that this is telophase is you see nuclear membrane starting to form again around the now 2 nuclei. So that's something else to visually say this is telophase. But this is the easiest thing to look for. Okay, that thing, which I said we're going to call something in a second, is called a cleavage furrow, right? What's a furrow? A furrow is like an indentation. A ditch along the side of a road is a furrow. It's commonly called a furrow, right? What does cleave mean? Cleave means to split, right? So this is a cleavage furrow, and if you see that, that means this cell is doing cytokinesis, but it also means it's in telophase, part of metaphase, right? This is actually already 2 cells. It's already been separated right here, and it's just beginning to separate here into 4 cells. You and I used to look like this at one early point in our life, just shortly after we were called a zygote. Okay, so here you see it again, this cleavage furrow splitting in. Again, if you're an animal cell, this is what happens. But if you're a plant cell, your cell wall is too thick and tough to pinch in like this. So this is how you accomplish cytokinesis. You have a whole bunch of vesicles that you make with your Golgi, right? And you move them to the middle of the cell. Your microtubules help that happen too. Remember, we've talked about microtubules helping vesicles move around. Here you see in an actual plant cell, here you see this line of vesicles. They're just going to continue to group up and merge with each other, and eventually they're going to hit the plasma membrane on either end, and now the separation of 2 cells will be complete. Each nucleus now totally separated from the other by its own plasma membrane. Around this plant cell is now 2 plant cells. Okay, so now we're to cancer. We'll do just a little bit with cancer, right? When things are working well, cells only divide when they're needed to divide, although the decision of need, again, is not a conscious one like we do, but it's due to some complex chemical stuff happening, right? But what cancer cells are, cells that might form a big clump called a tumor, therefore we could also call them tumor cells, right? What cancer cells are cells that just continually divide. They go from one mitosis cytokinesis to another, right? And any kind of cells can have this happen. So you have breast cancer, you have bone cancer, you have brain cancer. Any kind of cells can have this happen. Okay, I'm going to take you through these real fast. Okay, here we have a carrot cell that starts with 18 chromosomes. After mitosis, each of its daughter cells are going to have 18, not some kind of different number. Okay. Second one, diploid, we'll talk about that later. Two-end skin cells undergo mitosis to form other skin cells, not meiosis, having chromosome number same as they started with. If this was humans, that would be 46, and those skin cells would also have 46 after mitosis. Here we have multicellular, unicellular organism, right? In general, this is not a great question. In general, multicellular organisms like cheetahs reproduce sexually, right? And this paramecium, which is just one cell, uses asexual reproduction. And that asexual reproduction is going to be mitosis. So the cheetah doesn't use this, though. That's what prokaryotic cells do. The cheetah uses sexual reproduction, paramecium asexual. So that's the right one. Okay. Mitosis did not have cell division. We talked about this. You'd have two cells, right? No, you wouldn't have two cells, because if the cytoplasm didn't divide, you'd only have one cell. How many nuclei would it have? Two, and they'd be identical to each other. Mitosis happens without cell division, like in the muscle cells we talked about.